my primitive area. Because it's going to be this area that's going to monitor heartbeat, respiration, chemicals in the blood, you name it. It's monitoring everything that's going to be needed for homeostasis, the maintenance of the internal environment. As we look at this system, we mentioned in the spinal cord and the spinal nerves that we have gray matter, white matter. In the spinal cord, gray matter looks like that little butterfly, okay? White matter were all of those tracts, T-R-A-C-Ts, that would carry information up and down because we had ascending, descending. In the brain, we're going to see gray matter. This would be my area for neurosomas, neuro, nerve, soma, body. So therefore, nerve cell bodies. For the gray matter, they're dendrites and synapses that could occur. So what does that mean? My nerve cell body, my axon, the terminal ends, the dendrites. All right? So, this is, of course, the graphical representation that's easiest to think about for a nerve cell. This is not how all nerve cells are going to look, though, depending on what area they're in. But in the gray matter, this portion would be in the gray, and then this portion would be in the gray. Because a nerve cell is going to synapse onto another nerve cell, muscle, or gland. So if I've got my dendrites, nerve cell body, axon, terminal ends. The terminal ends of this axon could meet another axon. They could meet muscle. Think about the neuromuscular junction that you learned about with skeletal muscle, or a gland. Okay, so is everybody with me? So my gray matter, the nerve cell bodies, dendrites, synapses. When we're talking about the brain, this gray matter forms what's termed the cortex. So the cortex of the brain, it's this layer of the gray matter. It's over my cerebrum, my cerebellum, and then deeper within the brain, we're going to have areas of nuclei. Do you happen to remember how I referred to nuclei? What was our definition of nuclei? Or did we talk about it? We didn't? Okay. When we hear the term nuclei, we tend to think about nucleus of a cell. Okay? Well, in this case, for our central nervous system, when we hear the term nuclei, this is going to be an area that has a lot. It's a very dense area of nerve cell bodies. That's what that's going to mean. It's an area of nerve cell body. So the only part that's left now is my axon. The axons are making up the white matter. They're bundles of the axons, which we'll know as tracks. Wait a minute. Is this any different than what we talked about for the spinal cord? No, because my spinal cord had the gray matter, the white matter. And the white matter, like I was saying earlier, the gray matter was the little butterfly shape, okay? White matter were the tracks that carried information up and down, okay? Sensory information up, motor out. What's the difference now? Alex?
The positioning is the white matter on the outside of the spinal cord.
Well, if we think about it, in relationship to gray matter, once again, being nerve cell bodies, the white matter being tracks, the bundle of axons. Rather than information like the spinal cord doing this number, I'm now stuck here. How are tracks, these bundles of axons, going to run now? It's okay.
Down.
I got to run across. I got to go from left to right. I've got to communicate from one lobe of the cerebrum to another. I've got to make sure the cerebrum can communicate to the cerebellum, that we can communicate. Do you see where I'm going with this now? So now, rather than worrying about these tracks like we saw up and down the spinal cord, in the brain, these tracks now have to do this number, that number, do you see? Okay? And then has to be able to communicate with the tracks of the spinal cord. Does that make sense? Okay? So, once again, wonderful gray matter and white matter. Just like we saw in the last chapter, we have our coverings. We have those meninges. Does everybody remember dura, arachnoid, pia? Okay? Now, this time, let's take a peek at our brains. This time, look at the structure that we have. Okay? I've got this huge groove going down the middle of the two halves. Grooves. Okay? I've got foldings. I've got indentations. It looks like I've got nice separations between the lobes. If I look through here, it looks like I've got all this stuff still running once again into these areas. So the foldings of those meninges, they become a little more involved than what we saw with the spinal cord, because it's got a lot of tissue that we need to make sure stays covered and protected, all right? So, we're going to find that these membranes of the meninges, they're going to create these areas that are existing in the brain tissue. We have what's called a dural sinus. Okay? Dura mata. Okay? Sinus. Do you guys remember that term from part 1? Where it's like a empty space? Okay? So, they're creating spaces that wrapping is creating spaces to collect blood to help circulate through the brain. And you'll hear them referred to if something's wrong in that area. The dura mater, can fold, or it does, fold inward. Where I have this huge longitudinal fissure, it folds all the way in to the point where I can't do this, okay? But it'll fold in as far as it can to help separate the two halves. Okay? So that will fold inward. We call this area the falx cerebri, cerebri meaning cerebrum, okay? This fissure between the two halves, okay? Tentorium cerebelli. Which part of the brain are we referring to now? Cerebellum, okay? Cerebelli, cerebellum. Well, if you look at your model, you've got what looks like a separation between the cerebrum and the cerebellum. This is what we call the foldings for that area. The falx cerebelli would be what folds in between the two halves. We also get a subarachnoid space. Arachnoid mater, okay, just below it, we get this space that will separate that middle layer from the innermost, the pia mater. Because remember, dura was the outermost layer, arachnoid was middle, pia was the innermost. That means that the pia mater is basically, I wouldn't say stuck, but the pia mater is actually running every single groove, nook, cranny, everything of this tissue. Dura is the outermost one. All right? We get a subdural space where we get the arachnoid mater separated from the dura. So these wrappings, they get a tad bit more involved in the brain area than they did with the spinal cord. If you look at the structure that we get, this would be the bone encasing the brain. Very good protection for the brain tissue. And then as they're trying to show our layers that exist, with the pia, they're trying to let you see the pia is running every groove, nook, cranny, everything. This arachnoid area, the thing about the arachnoid area, the reason it's termed arachnoid is because it looks like a spider web. Arachnoid meaning spider, okay? And what that is vessels, blood vessels. Because the tissue of the central nervous system, the brain, the spinal cord, demands high amounts of blood. So the blood flow is great to this tissue. Every time your heart beats, I don't want to quote the number because I don't walk around with that number stuck in my head, but every time the heart beats, a certain percentage of that blood flow goes directly to this tissue to feed it. I do quite a few other things too. When we're looking at the structure that we have, a ventricle kind of brings a lot of things to mind about that. But with the ventricle, in our brain tissue itself,